Welcome to everybody, in particular to our speaker, Professor Ulanovsky. I will give some information about the speaker of today. We are very happy that he's here to discuss a very interesting topic about bats. And Professor Ulanovsky, after earning his BSc in physics from Tel Aviv University, he obtained his PhD in neural computation at the Hebrew University of Jerusalem. He worked at the University of Maryland, where he pioneered the tetrad recording techniques for recording place cells from the hippocampus of freely behaving echolocating bats. I hope I told correctly. Since 2007, he works at the Weizmann Institute of Science, where he is an assistant professor from the Department of Neurobiology of Weizmann Institute of Science, Rehovot, Israel. He obtained a very large startup allowance to begin his own lab with a tenure-track position. Currently, he is under review for tenure-track promotion. He is renowned for his work on the neural basis of spatial cognition, navigation, and spatial memory in the mammalian hippocampal formation, using bats as a novel animal model. He publishes consistently in all major journals: Science, Nature, the Journal of Neuroscience, Nature Neuroscience, Hippocampus, etc. His work was also featured by the Discovery Channel. He won an ERC grant and was recently awarded with the prestigious Bernard Katz Prize in Neuroscience, awarded by the Alexander von Humboldt Foundation. So he's an exceptional speaker for a very interesting and, I'd say, original argument. So please, Professor.
Okay, hello everyone, and thank you so much for the kind introduction and for the invitation. And do you guys at the back hear me? It's okay? Okay. What we're interested in is in understanding neural basis of spatial processing, if you will, spatial cognition, spatial memory, spatial behaviors in mammals. So it's kind of a broad topic, and you might wonder why space. And for me, there are several reasons. First of all, of course, it's important. Navigating or moving through space is important for all animals, so it's interesting in its own right. It also has some interesting, more general properties that space obeys some nice rules. So it's Euclidean, so you can define a distance in space, which is actually non-trivial, because if you think about other higher cognitive functions, so what's the distance between, love and hate, whatever, rage, and all of these things? It's more difficult to define. And here you can define a distance. You can also measure performance very well. How much of an error did an animal or a human make when going from point A to B? Or how straight did it go? So you can put numbers on the performance, and this immediately means that you can make models, as Alessandro here, for example, and many others are doing. So it's a very nice, I think, studying neural basis of spatial perception, spatial representation is a very nice system, more generally, in talking about higher brain functions. And the heroes, the brain regions that are the heroes of today's talk are the hippocampus and the entorhinal cortex primarily. So, so the hippocampus, this is in a rat brain. The hippocampus is this sausage, purple sausage here, and the entorhinal cortex is this dark purple band at the back of the brain. And these two brain areas are very important for spatial memory, memory more generally across mammals. And it's one of those great subcortical loops where information comes from neocortex, goes through the entorhinal cortex to the hippocampus, and then back to the entorhinal cortex and to the rest of neocortex. And if you go to these brain areas, you find very interesting neurons. And I'm sure many of you know about these, but I'll describe them nevertheless. So if you go into the hippocampus, you find neurons that are called place cells. So this shows an example of a place cell in a rat. So this rat was running in a box about 1 meter square. So the black line is the path of the rat, and the red dots are action potentials or spikes that were emitted by this one neuron. So you see that most of the action potentials are emitted in this northeast corner. You can also turn it into a heat map, And then you can see that this neuron likes to fire in the northeast corner. This is called the place field of this neuron for obvious reasons. It also fires a little bit here. And these neurons, for obvious reasons, were called place cells. Now, if you go one synapse back in this loop or one synapse forward, depends which direction of the loop you like better, you find neurons that are called grid cells. So these are neurons that now, again, you see the response of an individual neuron in a rat. This is from the Moser lab in Norway. And the rat is running again in a random path, more or less. And this one neuron is active in a set of locations when the rat is here, or here, or here, or here, or here. So you see that this set of locations form a hexagonal lattice or a grid. And this is autocorrelation of that picks up really nicely the periodicity in space. So this is very bizarre if you think about it. How come with a random walk of an animal you get a regular lattice that looks like something you'd expect to see from physics or chemistry, not so much from biology? So this is, I'll be talking about that. And you also find in the entorhinal cortex and some other surrounding brain regions, you find head-erection cells. So in the rat, again, an example of a neuron that does not care so much about where the animal is, but it cares about where its head is pointing. So in this case, when the head is pointing almost due east, the neuron fires a lot. This is kind of a polar representation of the activity of the neuron. And there are also border cells that I won't be talking about much, but if you think about it, this could be thought of representing a map, and the grid cells as well, the location of the animal, and the head erection cells sort of in neural compasses. So you have a map and a compass, and together you have all the components you need to navigate through space. However, we are studying these issues in a somewhat unusual animal model, the bat. We're in fact the only lab in the world who are doing these kinds of recordings in bats, and I'll actually leave to the end to justify why are we doing this in bats, because I think it's easier actually to first show the data and then become more clear why we chose the bat as our animal model. So I'll start with an introduction about the animals themselves. So bats are, first of all, mammals, and there are many of them. There are 1,250 species of bats, so almost a quarter of mammalian species are bats. Very, great variety of them. There are bats that eat insects. There are bats that eat nectar of flowers. Some eat fruits, like the bats that we are studying. Small vertebrates like frogs, and even fish that these guys in Central America and South America can take off from underwater, because as the fish, in complete darkness, as the fish is approaching the water surface, it's still below the water surface, but there are concentric rings that are formed on the water surface, and then the bat, with using its sense of sonar, identifies this and puts its claws in the middle of the ring and can take the fish outside, although it's still underwater. So it's all in complete darkness using its sense of sonar or echolocation. And this is the bat that we are working on now in my lab at the Weizmann Institute. It's the Egyptian fruit bat. It's actually, first of all, you'll immediately notice the large eyes. So these guys see very well. In fact, all bat species see. There's this saying, blind as a bat. It's not true. All bats see. These guys see really well. Their visual acuity is much better than rats. And they also have this sense of sonar. They're also very large, so compared to bats at least, other bats at least. So the bat I worked on during my postdoc, the big brown bat weighed about 15 grams. This guy is 10 times larger, so we can put a lot of equipment on it. Neural telemetry systems, neural loggers, GPS data loggers, things that I'll be talking as we go. And so this kind of a low-tech solution to the miniaturization problem. Just use an animal that's 10 times larger. And they're also very nice animals to work with. So what we do routinely in the lab as we run an experiment, the two things we routinely measure behaviorally are the position of the animal in 3D space. So we use two cameras that are synchronized, and then you can reconstruct the three-dimensional position of the animal. And we can also record with microphones the sequence of echolocation clicks. And less routinely, we measure where is the animal directing its beam in space. So we do that by using an array of microphones. We did that. We had several studies on that with an array of microphones, also microphones on the fourth wall. And this is how an emitted call looks. So it's intense in one direction and falls off in other directions. And to show you what kind of data you can, the behavioral of the bat, I'll show you a short movie where the bat will fly to land on this target here, just a big styrofoam ball. And here you'll see, these are the top view reconstruction, these are the microphones, and the bat will be flying like this to the target which is right here. Oops. So what you'll see immediately, okay, the bat is flying over there. The bat is directing its beam very repetitively in space. Left, right, right, left, left, right, right, left. And as it approaches the target, you'll see it does not look straight at the target, look acoustically, but looking to the sides of the target. Okay, so as if it's really trying hard not to look straight at the target. And why are they doing this? Again, I will not go into the details, but the rationale of that is that what we showed in a series of studies is that if you look straight at the target, this is best for detection because this maximizes signal to noise. You get the strongest possible echoes because this is largest intensity. But this is worst for localization because this is zero derivative of the emission beam, and then any movement, relative movement in the angle between the bat and the target would not change the intensity of returning echoes. So this is not good for localization. What is better for localization is to look sideways and put this point, the maximal slope, onto the target. And this is the most sensitive region to any changes. And this is exactly what we showed that the bats are doing. They're putting exactly the maximal slope onto the target and optimizing localization this way. Okay, so this was kind of an introduction, a bit of put you into the world of bats, if you will. And what I will be telling you about today are several stories. I'll be telling you about GPS tracking of bats in the wild. So this is behavioral studies of how bats navigate in the wild. Also about 2D grid cells in 2 dimensions without theta oscillations. I'll tell you about 3D place cells, 3D head direction cells, and maybe even 3D grid cells. A little bit about future plans. And if at any point you have questions, I don't know if it's customary to stop, but I'm perfectly fine with that. I'll be happy to answer in the middle at any point you want. All right, so I'll start with the first study, GPS tracking of bats and bat navigation in the wild. This is the work done by Asaf Tzor in collaboration with Ran Natan from the Hebrew University. So the basic question that we asked here is, how do mammals navigate? I mean, really navigate in the wild. And you'd think that this is a question that we must know the answer to by now, but in fact, very little is known about this. There's a lot of research done on navigation of birds in the wild, and of insects and Pacific salmon and sea turtles and lobsters, a lot of creatures, how they navigate in the wild. And you'll notice that all of these are not mammals, meaning the brain structure is quite different, and also very little is known about brain mechanisms in these animals. So this comes from the tradition of ethology, European tradition. And then there is a very different approach coming from psychology, where people have been running rats in mazes for the last 100 years, and there people have found these place cells, grid cells, head direction cells, but this is all in very small environments, very artificial conditions, very different from the very small scale, very different conditions in the real world. So these are two worlds that are two approaches that really don't talk to each other much, and eventually I'd like to bridge the gap between the two. So as a first step, we just asked the behavioral question: how do mammals, and bats in this case, navigate in the wild? And so for that, so this was kind of to bridge the gap between the ethological approach and the psychological approach. And for that, we use these tiny GPS data loggers that store the position of the animal on board this logger. Now we have a smaller version of it made by an Israeli company, and you put it on you glue it with a medical skin glue on the back of the bat, and release it at the cave. So it's literally not wild type, but wild bats that we catch in the wild as they're exiting the cave. We put this device, this logger device, and then we release them. And what you'll see now in a movie of a flight of one such bat. So the red line is the path of the animal. The illumination is wrong because Google Earth gives you daylight illumination, but of course they fly at night, so just imagine this is dark. But the path of the animal and the topography is correct. And this is a bat so this is the cave, okay, so it's about 40 minutes drive from the Weizmann Institute, and the bat leaves the cave and starts flying west, almost west. This is here the Mediterranean Sea. And it flies west for a very long distance. This is about 15 kilometers up to that point. And when something interesting happens, you'll see that it looks like it's keeping more or less fixed altitude. And here it passes this big highway. And then at some point, this particular bat did something interesting. You see that it kind of veered left and then started flying along the backyards of this little village, Moshav. Until it reached this tree right here, this fruit tree. It fed there for several hours and then eventually flew onwards. Now this was the only bat that did that, only individual bat. All the other dozens of bats that we tracked did not fly along roads or power lines or anything like that, but they just flew straight. So this is a more representative example of another bat that was again captured in the same cave. And released with the GPS data logger, so it flew to a local tree for just sitting there for a few minutes, and then flew very straight to these two trees, fed there for the entire night, and then flew back. And note the scale here, very, very, very straight, homing back. So these guys can really navigate. And so, to summarize the kind of the results from many such bats that were tagged. We see that they fly very far, very straight, very fast, very high, and they return to the same individual tree night after night. So, so this is an example of an individual bat that was tracked for seven nights back and forth. So it's going from the cave to its favorite fruit tree and back. So there is some spread here, of course. But they're navigating very well, and they're returning again. With the GPS, we have enough resolution to say that they're returning to the same individual tree night after night, which means they're not just randomly flying straight, but they're relying on spatial memory. They're really navigating very well. And again, I don't have time to tell you about all the details, but what we've done a whole series of translocations where we basically try to replicate at least the mode of thought of people who do laboratory experiments. Your classical experiment to see if they have a map or a map-like representation of spaces, that you do shortcuts experiment. You translocate them somewhere else and see if they can do novel shortcuts. And this is what we did, but on kind of in real environment outdoors. And so we released them in several locations south of the Weizmann Institute, the most extreme of which was in the middle of the desert where bats don't live, in the middle of nowhere, inside. So we asked, do they use vision for navigation? So we released them inside a big erosional crater about 300 meters deep. From there, there's nothing familiar that they can see. And these bats indeed basically got lost. And eventually they went out of the crater and flew north. They were able to see what we think the lights of the Mediterranean Sea, and they flew north to their cave. Another group of bats were released on a high mountain right next to the crater, and from there you can see the Mediterranean Sea, Tel Aviv lights, etc., and they flew just straight. So there was this very dramatic difference, very nearby locations. Once in the hole got lost, once up there flew straight. So we think it means they use vision and possibly very distant landmarks like mountains, the sea, cities, etc., to navigate. This is kind of a brief introduction, just to say that these bats are really excellent navigators. It will be interesting to see kind of the neural basis of these navigational abilities eventually. Okay, so going from natural scale experiment to lab experiment, which is most of what we're doing. I'll start with two-dimensional experiments. So about grid cells without theta oscillation. So this is work done by a fantastic PhD student, or a former PhD student, Michael Yartsev, in collaboration with Menno Witter, a noted neuroanatomist from Norway. So the question that we asked again is how do you get this grid-like structure? And there were several models proposed to that. Santo proposed one nice model, and there were additional two classes of models. One class that relies on network interactions. So without going to the details of this, these models propose that you have a Mexican hat connectivity, circular symmetric connectivity in the network, and then the minimal energy of the network will be such that the blobs of activity on the network will be equal distance from each other, which is exactly a hexagonal arrangement, and then with the right synaptic connections, the activity will follow the movement of the animal, and then you'll get on an individual neuron basis an activity that looks like a hexagon. Okay, so this is kind of one set of models. And another set of models rely on oscillations, temporal oscillations. So the idea there is you have an oscillation in time called the theta oscillation that runs in the soma of the neuron, and then you have an oscillation in the dendrite that runs at a different frequency. And the assumption of this model is this difference in frequencies is proportional to the running velocity of the animal. And why would that be useful? Because now and then of course you'll get beats like this, and if now the animal runs twice as fast, then the frequency of the beats will be double, the period of the beats will be half, but because the animal runs twice as fast, the wavelength in space will be fixed. Okay, so by this assumption of frequency difference being proportional to velocity, you essentially integrated movement, and you get oscillation in space, if you will, that is has a fixed wavelength regardless of the running speed. Of course, time is one-dimensional, so you will not get something like that. You will get maybe bands, and then you need to assume that you have three inputs at 60 degrees to each other, and then you get those grids. So this is kind of the essence of these kind of models. And the problem is that it's very difficult under normal conditions in rodents. It's very difficult to dissociate these models. So why is that? Because you always see those grid cells together with those oscillations. So it's just difficult empirically to dissociate the two. And here bats come in handy. And why is that? Because during my postdoc, when I worked with the big brown bats, I found place cells. And here you see place cells from our bats, Egyptian fruit bat from the lab at the Weizmann Institute. So recording from the hippocampus. And you see here 15 cells. This cell likes the northwest corner, and this is the southeast corner, etc. So they kind of span the environment. So this is, and this is in two dimensions, yes. So in the box, essentially, you kind of ratify the bat, let it crawl in a flat box with a white cue card, on purpose, very similar to the way you do an experiment in a rat. So everything is very similar to a rat. However, the main difference was in the theta oscillation. So unlike the rat, where as it's running around, you see all the time this 8 hertz low frequency oscillation. It's running continuously, essentially, in the rat or a mouse. In the bat, you see something very different. You see these short bouts that last maybe 1 second, typically, and then nothing for half a minute, and then another bout of theta, and then nothing for half a minute. So very intermittent. And something similar, by the way, was reported also in humans and in monkeys. So it's very difficult in primates to find this kind of continuous oscillation. So this looks very different than in the rat. And of course, it immediately raised the question of whether an oscillation like that could be used to produce the grids. And this is indeed was our rationale. We asked basically if in rodents it's difficult to do this dissociation, at least under normal conditions, in the bat we kind of, for free if you will, might get this dissociation. Because if in the bat we go one synapse back into the entorhinal cortex and find grid cells like these, but without the theta oscillations, then we kind of argue against this class of models. And that's indeed what we did. And I'm jumping to the next slide. In between was a couple of years of work just to find the brain area and et cetera. And we got a lot of help from Menno Witter, a neuroanatomist who helped us define the brain area in the bat. But eventually we were able to find the area and find grid cells. So this is an example of the grid cell recorded in the medial entorhinal cortex of a bat. And we found also that many of the properties were very similar to the rat. So the angles, this is again data from the bat, the angles were on average 60 degrees. We had didn't have many examples, but we had a couple of examples of several grid cells recorded simultaneously. So here you see 4 grid cells recorded simultaneously and showing their preferred orientation. So this angle. and you see that this is on a 60-degree cycle of a hexagon, and you see that the orientations tend to cluster together much more than expected from chance. This is similar to what you find in rats, whereas the spatial offsets or spatial phase is kind of random. So if you look at the spatial cross correlations of these same four cells, you see that the central blob is above the center here, and it's below center there, and it's on center here. So kind of the spatial phase is all over the place, again, as in rats. And finally, we saw this interesting gradient where if you look at the grid spacing or the wavelength of the grid versus the physical position in the brain, so literally dorsoventral position in the brain, you see that dorsally you get smaller grids, and it increases, the wavelength increases as you move more ventrally. Again, it's something similar to what you see in rats. So the reason I told you about all these detailed properties is to say that grid cells are not just there, but also many of their properties are similar to what you see in rats. And we also find some of the other cell types. We had a few border cells and some heteroaction cells in the internal cortex, again, similar to what you find in the rat. So functional properties of neurons in this brain area looks quite similar to the rat. What about the oscillation? So again, this is in the rodent, the mouse in this case. If you move your recording tetrodes into the internal cortex, which is this brownish area, you start picking up very strong oscillation. This is the power spectrum of the local field potential, and you see this huge peak in the spectrum at around 8 Hertz. This is this theta oscillation in the mouse, which is very strong. If we do the same in the bat, we get nothing. We get this 1 over F dependence, this generic dependence, no peak at 8 Hertz. What about autocorrelations of spike trains? This is again in the mouse. You see this very clear rhythmicity in the autocorrelations, whereas in the bat we don't see anything of that sort in single units or in multi-units. And we quantify this by looking at what people have used before, the theta index, which is essentially how much energy you have in the autocorrelation and in the theta frequency range. And by shuffling the spikes, you can compute confidence intervals, and you see that pretty much none of the neurons or the multi-unit clusters did not go out of the confidence intervals, meaning there is no significant rhythmicity in the firing of these neurons. And we've repeated this now. I don't have the slides here, but we've repeated this now for other frequencies, because some of the arguments that people said, maybe in the bat it just does not work at 8 Hertz, it works some other frequency, and there's some reasons for suggesting that. So we now, in an unbiased manner, look at 1.5 Hertz, 2 Hertz, 2.5 Hertz, every Hertz index across grid cells, place cells, and interneurons in crawling and in flight. I'll tell you about the flight data soon, and we don't see pretty much any significant oscillation or anything consistent across those data sets. So I think it is more than just showing that it's not a theta, but it does no movement related oscillations, at least in the bat in any frequency. And finally, we asked here whether those short theta bouts that I've showed you, they're rare, but maybe they still contribute somehow to setting up the grid. And so what we did here is we computed the firing rate map and the autocorrelation, and then we identified those theta bouts and removed them. Okay, so you see this is before removal, this is after removal. It looks pretty similar, but there is some numerical difference in this gridness index that is customarily used for assessing the hexagonal symmetry. However, of course, this difference could be because you removed some amount of data. So to assess this, we plot now the change in gridness index, so the difference between this and that number. But for computing the confidence intervals, we now delete the same amount of data, but unrelated to the bouts. Okay, and we repeat it many times, and then you can compute the confidence intervals, and you see that none of the neurons, none of the grid cells, was the real data outside of the confidence intervals, meaning that really those theta bouts do not contribute to setting up the grids. Okay, so taking all these data together, this suggests that this argues strongly against this class of models, right? Because this really, in a causal way, because this model suggests you need A to get B. You know, oscillation time to get an oscillation in space, we show B without A, so this really causally argues against this kind of models. All right, so this was a story about 2D grid cells. Now I want to move to 3D flying bats, which is an obvious question to ask in these animals. This is again work done by Michael Yartsev. And so this is kind of the question that we asked, right? This is how grid cells look in 2D, maybe. You can think about this way. Would it look something like that in 3D or somehow else? There are all sorts of suggestions. Or you can ask the same questions about place cells, right? When you see a two-dimensional place cell that look, the firing of which look like that, you could imagine that in 3D it will be any of those, right? And you really don't know. Or if you see a place cell that has two fields, which you sometimes do in bats and rats as well, you can imagine that in reality you have these floating balls in midair, but you just happen to put your floor like this, so you see these two activity blobs. So you can make a lot of pretty plots, but you really don't know until you actually do the experiment. And people have done experiments in this direction. So there were experiments with, from the McNaughton lab where they sent rats to space on board a NASA space shuttle with tetrads in the hippocampus, and they had rats trained to run along this Escher staircase in microgravity in outer space, and experiments on planet Earth in London. In Kate Jeffries' lab, had rats running on a vertical wall, and there was another variety. But I would argue that if you want to ask the questions about representation of three-dimensional volumetric space, this does not answer this, because this is essentially a one-dimensional path embedded in 3D. And this is a wall, a two-dimensional wall. It's vertical, but still two-dimensional. So really, to answer the 3D volumetric coding question, you need to have animals going in 3D space. So you could do this, in fact, I think with rats, if instead of these kind of pegs, you have this 3D interlaced array of rods, and some people are trying to do this now in rats, but for that then you need, of course, to get rid of this cable that will create a problem in such a setup. We've done the same approach, so getting rid of cables and do it in flying bats that, of course, move volumetrically very well. And we had to solve two problems to do that. One is to be able to record in freely flying animals, which is required developing a telemetry device, a neural telemetry device, which together with Neuralinks, and it took us several years, but eventually we were able to record spikes. So here you see the 4 channels of 1 tetrad, and you see they can record nice spikes. This is in flight. And the other problem that we had to solve is behavior, which sounds maybe easier, but as those of you who are doing behavior actually know, sometimes behavior is more difficult than electronics. So how do you convince an animal to fly in 3D and then fill in the volume very well? Because initially we started by just putting 3 targets in the room, and they just very quickly solved the traveling salesman problem and just were doing one-dimensional flight trajectories in midair, which is not very good for us. And then what happened is that I saw for the first time these GPS data. So I've showed you these long distance flights, but in fact, if you from the cave to the tree, but if you zoom in around the tree, this is what you see. So this is real data, and you see that the bat is flying around and around and around the tree, covering volume very well. And so I saw this, that this is basically what we want to reproduce in the lab. So this is what we did. We built this artificial tree, or a 3D 8R maze if you're a psychologist, same thing. And then basically what we do is we randomly put food on the arms, so the bat does not know in advance where the food will be, and basically has to fly around and look for where the food is. And the tree is placed in the middle of the flight room. We have a flight room which is almost 5 by 6 meters inside, and again the same 2 cameras that allows it to reconstruct the 3D movement of the animal. And indeed this convinced the animals to fill in space quite well. So here you see a flight path of a bat. From a top view, of course, you get a hole in the middle, but from the side views, you see the coverage is pretty good. and they fly sometimes more than a kilometer. Now we have up to 2 kilometers, so they fly in one session, so they fly pretty well. We now found that there is a way to get rid of this hole, which will be not very good for grid cells, because in fact if you, instead of the tree, just put many targets in the room and randomly put, without a tree, without this pole in the middle, but just put targets that hang from the ground, from the ceiling, 6 or 7 targets, and randomly put food, it essentially amounts to the same thing. They basically don't know where the target is and they just randomly search for it. So we are able now to get rid of this hole in the middle. But in any case, if we now connect these two together, connect the flight paths of the animal to the spikes that we recorded in the telemetry system, we get something that looks like that. All of this is one cell, one neuron in the hippocampus C1 region and dorsal hippocampus of the bat. And what you see here is that this neuron is firing in a spatially localized manner, including the height. Okay, so it's also at a certain height. So here you see the top view and the 2 side views. These are kind of rate maps, and this is a 3-dimensional view of that. And these are 2-dimensional cuts. So essentially, if you look at this volumetric firing rate map and you start doing these 2-dimensional cuts going from the left wall rightward, so like this. So in the beginning there is nothing, and then as you reach 3 meters from the left wall, so about here, you see that there is activity, and then it kind of fades away as you go more to the right. So these are just various ways of looking at the 3D data and showing that indeed you have a localized activity in space. And this is another example of another neuron which we recorded in a different shaped enclosure. This is a cubic enclosure, and again you see this particular neuron is active about half a meter above the ground. So we recorded in total 55 of these neurons, and what we see is that, first of all, and again I won't go into the details because I want to move to newer data that was unpublished. We see that first of all, most neurons are isotropic in the sense that their place fields are not significantly different from a sphere. So meaning the resolution is the same in the X, Y, and Z directions. And also we see that the place fields are reasonably uniformly covering space. So we don't have enough neurons to really tell for sure if that's the case. But if we arrange the centers of mass for the X, Y, and Z directions separately, see that it kind of covers all possible positions more or less uniformly. So roughly, at least as much as we can tell, it seems to be covering space pretty uniformly. And finally, as I said, we looked at the theta oscillation, again asking a question, maybe in flight it's somehow different, and the answer is no. There's still no, again, we do look at the theta index for all the cells, and there are almost no cells that are significantly rhythmic in their firing, in the spike trains. Also in flight, despite several reasons why you could have expected perhaps theta to exist in the flight, but at least we don't see that. And as I said, we now looked at other frequencies as well, like basically all frequencies in an unbiased manner, and also interneurons, which is very important because interneurons are in rats are known as theta cells because in fact they're even more strongly oscillatory in the rat than the place cells are, and they also have very high firing rates. It's easy to detect oscillations if they are there, but we very clearly do not see oscillations in the interneurons in flight or in crawling. Okay, so far published data. I'll now move to unpublished newer results. So this story is about 3D head direction cells. So this is work done by a fantastic PhD student, Arseny Finkelstein, and with a collaboration with Dori Derdikman, who's now in Technion. And again, the question that we asked is essentially, do you have 3D compasses in the bat's brain? So we know that you have, as I showed you already, you have these head direction cells in the rat that like to fire a certain azimuthal direction, and we found them in the bats as well. This is a cell that's active in a certain azimuthal direction in the bat. What about 3D? So to answer this question, we came up with this kind of device. So this is a non-coplanar array of LEDs that you photograph with one camera, and then because they're non-coplanar, it means that let's say if the animal looks up, then these green LEDs move backwards in the camera view, as opposed to these red and blue LEDs. If it looks down, it's the other way around. So with few pages of trigonometry, you can reconstruct all the 3 Euler angles, yaw, pitch, and roll, or azimuth, elevation, and roll, from view for one camera. And then so we can measure all the three Euler angles of the head, and while recording neurons in the brain. And the brain area we chose to record from is the presubicculum, which is this area here, which is actually the first brain area where heteroaction cells were discovered in rats by Jim Rank and Jeff Taubey in the 80s and late 80s. So so when we looked at the data that we collect, we see a variety of cells. So first of all, we see cells that look like that. So what you see here are two sessions. This is one session, this is another session, separated by an hour. So this is the point of that, is just to see that this is similar to that. So you have stability over an hour. And what you see here is the firing rate, the activity of the neuron in color-coded. So red is high activity, blue is low activity, for elevation versus azimuth. So you see that this neuron doesn't care so much about elevation, but it's very strongly tuned to azimuth. So it likes 360 degree or 0 degree azimuth. Okay, so this is your classical azimuthal two-dimensional rat-like heteroaction cell. Okay, but we also see neurons that look like that. This neuron does not care so much about azimuth, but it cares about elevation. So it's, when the animal is looking down, in this case, this neuron fires strongly. And we also see neurons that look like that or like that. So they're conjunctive. They care about a combination of azimuth and elevation. And we also see, rarely, don't have many of those, but sometimes we see neurons that look like that are triply conjunctive. So they like a combination of a certain azimuth and elevation, but also roll in azimuth and a roll elevation. So they're tuned to all three Euler angles. And there are neurons were pretty common. About 30 of the azimuth-tuned cells, about 35% were also tuned to elevation or roll. So this is a sizable fraction of the neurons. So first of all, yes, bats do have 3D neural compasses in their brains. We also found this very intriguing in my mind, functional gradient. So if you look at, this is sagittal section of through the presubicculum, this is the hippocampus here. So if you go now away from, so this is the subiculum, away from the hippocampus, and just ask what is the fraction of these various neural classes, you find that around 0 here, close to the subiculum, 100% of the cells are pure azimuth cells. So very clearly, almost only pure azimuth cells here. And as you go backwards along this line, it's actually a bit more the diagonal line in the brain, but it's more difficult to depict. As you go away from the subiculum, along the transverse axis of the presubiculum, you find that this fraction goes down to about 35%, and all these other neurons, three-dimensional conjunctive and elevation neurons, go up to 65 or 70%. So it's literally as if going away from the subiculum, you move from a two-dimensional rat-like representation here to a three-dimensional bat-like representation over there. And I think these tendencies are very clear. I'm not entirely sure what it means, but at least there's this very clear functional arrangement. And in fact, this will predict, because the transverse, for the aficionados here, that there are anatomical studies that show that the transverse axis of the presubiculum maps in an inverse manner into the transverse axis of medial internal cortex. This will have implications for coding of perhaps of vertical dimension in grid cells along the transverse axis, which is the one axis perpendicular to this axis where the lambda, the wavelength, increases. So the other axis, you'll have maybe a differential representation of height. Okay, so we have this interesting functional gradient. And lastly, we asked what happens in an inverted animal. Of course, bats like to spend a lot of their time inverted. Mice, by the way, do this as well. So rats don't like being inverted too much, but mice like a lot. If you go into a rack of mice cages, I don't know if there are any mice people here, but just go into a rack of mice cages, you'll see a quarter of the mice or whatever are just hanging upside down, just like rats. So mice are really good at that. So it's not just a bad thing. But in any case, we asked what happens in an inverted situation. So what we did is we had 3 sessions now: an upright session, an inverted session, the bat was inverted here, or an upright session again. And here you see examples of 3 neurons. These are the 3 columns. And so you see that in this cell, for example, in the upright session, it was tuned, and in the second upright session, it was tuned again with the same angle. So just show you that there is stability. But in between, this neuron was still tuned, but to another angle. And if you look at this neuron, same thing. It's still tuned, but to another angle. Still tuned, but to another angle. And it takes most physicists about 7 seconds to figure out what's going on here. It took us 3 months. But I think Sarah Soya was the world record holder. She'd figure out in 3 seconds what's going on. Said, oh, it's 180-degree shift. So indeed, that's what's going on. We have a 180-degree shift here in these 3 examples. So you shift the tuning curve by 180 degrees, aligned exactly under the original. And if it's not just these 3 neurons, if you look across the neurons, then you see, again, if you compare the 2 upright sessions, the difference in the preferred angles around 0, which just means stability. But if you compare the difference in angle between the upright and inverted, you see that you have this U-shaped distribution with peaks at plus minus 180 degrees, which means you have this 180-degree shift or inversion. So what does this mean? We think that this means is that the 3D head directions are represented not in spherical, kind of your textbook classical spherical coordinates, but in toroidal coordinates. So in toroidal coordinates, you have 360 degrees of azimuth, but also 360 degrees of elevation. And the rationale for, and so this basically means that in toroidal coordinates, you don't have any flip of 180 degrees because all of this is the same azimuth. And what would be the advantage of that? The problem with standard spherical coordinates is that you have singularity points at the plus and minus 90 degrees. So if you imagine that, you know, I'm a neuron, azimuth tuned neuron that likes, you know, that direction, and now the animal moves its head like this, so the neuron still fires, and then, you know, I'm at 89 degrees, then I move to 91 degrees, then the neuron is supposed to stop firing in a very abrupt manner, right? So there is this singularity point there, and same in the minus 90 degrees. So singularity that seems, you know, unbiological. Neurons are not supposed to stop or start firing in such an abrupt manner. Whereas in a torus, this problem is solved. The torus, you have a continuous angles there. And so indeed, when we compare the correlations between the upright and inverted maps, assuming spherical coordinates versus toroidal coordinates, the data is more consistent with a torus. So we think that this is evidence for a toroidal coordinate system. And now, of course, I'm slightly cheating you because, of course, there's no magic. 3D angles are always tricky, and the trick here is that you kind of ignore the role, right? We basically put the problem elsewhere. So we now looked at two continuous angles, azimuth and elevation, but what about role? We essentially binarized the role. It's now upright or inverted. And is it justified to do something like that? And we argue that yes, and for 2 main reasons. First of all, neurally, the neurons tend to be much less strongly tuned to roll than to azimuth or elevation. And the tuning tends to be less stable. So it seems to be represented by the neurons less strongly. And the other is the behavioral arguments, that bats do a lot of azimuth maneuvering, and they'll do a lot of elevation or pitch maneuvering, but they hardly ever roll. And to convince you that that's indeed the case, I'll show you a little movie that we took just bats landing on the ceiling, on a grid in the ceiling. And so this is the grid, and you'll see that the bat is doing a 180-degree elevation flip or a pitch flip here, clear here. Right there it went to the plus 90-degree elevation point. See, right there, plus 90 degrees. So it's not just a rare thing that they have to go through these singularity points. They go through these points all the time. Right there went to the plus 90-degree point, okay? And also in taking off, they go through the minus 90-degree point, right? So you see right there it went to the minus 90-degree point, okay? So this is a problem they have to solve in real life. And so they do a lot of these pitch maneuvering, but they, and as you'll see in a second, they do also a lot of azimuth maneuvering, but they hardly ever roll. So this is a bat that flies. Okay, you see that now it will do 180-degree azimuth maneuvering. This is slowed down 10 times, but you see that the head is almost level. They don't roll very much, okay? The head is almost level, and here the bat will gain elevation, but the head will almost not roll. Okay, so we looked at a lot of these movies, and it's also other people have looked at that. And flying bats, they don't seem to roll much. So that's why we think it's justified in the torus model. And finally, we asked what happens in flight. So all that I showed you is in crawling bats, right? So is in flight, is it different or the same in flight? And so very recently, Arseny has recorded from the same neurons in the crawling task and in flight using this neural logger I'll show you in a second. And you see that this is an example of the same neuron recorded in flight and crawling, and you see that the azimuth is different, which is of course expected because these are actually two different rooms. One is the flight room and one is the crawling room. But interestingly, this example, and we see a few more examples, we now try to look at whether it's systematic. It looks like the pitch tuning or the elevation tuning is the same. So it likes negative pitches in both conditions, which is actually something you might expect because you do have a geophysical axis of pitch, which is gravity, right? So you could sense gravity the same in both rooms. So it'll be interesting to see whether this is the pattern that's kept across neurons. So Arseny is literally spike sorting as we speak, so we'll hopefully have an answer to that soon. He recorded quite a lot of data with this conditions. So kind of as an interim conclusion about 3D spatial representation, we do see 3D place cells. The representation of 3D spatial representation is uniform, is isotropic, and is based on a rate code in the sense that we don't see an oscillatory temporal code in the firing of these neurons. And 3D head erection cells, we do see them, so there are 3D neural complexes, and we argue that representation is toroidal. And as a bit of speculation or a teaser, I would like to argue or propose that this is something that should be looked at in other mammals. So of course you could think of bats and dolphins that are obviously 3D animals, but also squirrels or rodents, like squirrels that jump between branches, or monkeys or cats. Okay, the jump between branches, you should have some sort of representation of 3D, and there's very little known about that, and I think it should be looked at. Okay, in the remaining few minutes, I'll just show you one slide about where we are with the search for grid cells, and then we'll summarize. So, so this is a project that is being led by Gili Ginosar, a new student in the lab, and with the participation of Arseny Finkelstein as well. And again, this is the question, right? What do you see in 3D? And we don't have a clear answer yet, but what we do have is already a lot of neurons that look like that. So these are neurons that recorded. We have one bat in medial entorhinal cortex and another bat in presubiculum in flight. So in both of these brain areas, you find grid cells in two-dimensional grid cells in rats. And the activity of these neurons looks very different than what you see in CA1 in hippocampus. In hippocampus, either you see one or two blobs, place fields, or if they're non-spatially tuned, then their firing is ugly. I guess that's the best word you can describe. Nothing systematic. But in entorhinal cortex and presubiculum, we find a lot of these neurons that look like that we'd never see in the hippocampus, which are kind of, I call them here blobby cell until we decide exactly if they're grid cells or not. But it's very clear, kind of distinct blobs. And we are now looking at actually using very helpful advice that Federico there gave us about how he analyzes kind of 3D grids. So we're now looking at 3D autocorrelations and at various kind of cut planes through these autocorrelations, and we see all sorts of interesting hexagonal structures, etc. So we're trying to figure out what's going on, but we definitely see irregularity in some of these neurons. So we still need to convince ourselves this is real, but it looks like some of these neurons at least have this spatial regularity. Okay, so let me summarize just by saying a few words about future plans where we're heading. So one is optogenetics. So we have some various ideas about looking more mechanistically at these issues, and we've been working in collaboration with the lab of Ofer Izhar in our department to develop methods for optogenetics. And so far we've only done the first step, which is actually, at least for us, was the most non-obvious, which is to see that the virus works. So we find that AAV5 viruses work very nicely in bats. So at least this is very encouraging. So we're now moving forward with that. And the other direction, which is very challenging, is to answer this kind of question. Okay, so as I said in the beginning, all of these experiments, starting with O'Keefe were done in small laboratory environments, and maybe a bit larger laboratory environments, but still very small scale compared to something like that. So the question is, do you get the same neural codes when you go scale up to, let's say, 1 kilometer? And it's not just an exercise in comparative experiments, but there are concrete theoretical predictions that on very large scales the code will be completely different. For example, place cells, according to Ketch and Zhang's models, might have dozens of fields. So it's as if a V1 neuron will have dozens of receptive fields. And there are also grid, Lafitte and others had ideas about grid cells having combinatorial grid codes that are, again, very different from what you might think otherwise. So there are concrete suggestions for very different codes when you go to a very large scale, and we want to test that. And of course, to be able to answer a question like this, you need to be able to record on this kind of scale. Here, our telemetry device will not work. It only works a few tenths of meters in the laboratory, so we really have to record the data on board the animal. So we've been working, developed this miniaturized neural loggers, devices that store the data on top of the animal in collaboration with a small Israeli company. And it's been already working for a few months now in the lab. We recorded with 2 or 3 bats already with this device. So this is how it looks on the bat. It plugs into our regular tetrode micro drive and the various views of that. And you can get really nice data with this. So you see here kind of clusters. If you do like your peak, your standard tetrode plot, you see this cluster from one neuron, another neuron, a third neuron, and we have actually a much better example by now. We should probably replace them. So you can get really nice data, and so hopefully in the next year or two we'll be able to start answering these questions about 1 kilometer of place cells. Do they exist and how do they look? Okay, to summarize, and this is my last slide, as I promised in the beginning, why do we use bats? So the question that I want to stress, the question, we're not after understanding bats. I mean, I think they're amazing animals, cute and very interesting to understand, but really after understanding the brain. And I think that bats are a useful model to answer questions that are difficult to answer using rats or mice. So for example, they have these temporally discrete sensory inputs, right? You put a microphone and record at a millisecond precision when did a bat get its sensory inputs. It's something that in a rat, as it's kind of running in a maze, when did the rat see something, touch something, sniff something? I mean, it's maybe doable, but it's very difficult, let's say. And in a bat, just put a microphone, it's very easy to do. And I didn't show you that, but I found that there was very interesting dynamics around this echolocation call. So immediately after an echolocation call, when all the rich echoes arrive, you have a place field is small, and then within a few hundred milliseconds, it diffuses a little bit. And then new call arrives, new information arrives, it shrinks back, and then diffuses, shrinks back, and diffuses. It's a little bit like the dynamics of ellipses of uncertainty in robotic navigational algorithms, but you literally see it in the place cells. You can ask questions, of course, about 3D. Theta oscillation happens to be different. Wasn't expected to start with, but this is the finding. So it puts constraints on computational theories of hippocampal neuronal function. They have excellent spatial memory, and which allows to hopefully try to bridge the gap between laboratory scale studies and natural scale studies using bats. And the 2 most important points for me are the following 2. So the first is the notion of comparative systems neuroscience. So in sensory systems, particularly in vision and audition, there's been a lot of comparative work. There's been at least probably 15 species studied in great detail, V1 in functional organization and activity of neurons in V1, so we can see what's general and what's not across mammals. In hippocampus, the situation is very different. 99.9% of what we know about the hippocampus physiologically comes from just one species, the rat, and a little bit from the mouse, which is phylogenetically very close. And so I think that, at least as a sanity check, we need to look at other species like bats and others to see what's general and what's not. And I think it's the invariant properties that we should focus, we as a community should focus on. And the other point is the issue of natural neuroscience. I really believe that and it's not just belief, there's not a lot, but there's some evidence that every time you go more in the direction of the natural behavior of the animal or take into consideration the natural behavior of the animal, you find new and surprising things about the brain. So I think this is the direction that neuroscience in general, and certainly our particular research in our group is going to go in. Okay, so finally, I'd like to thank lab members and alumni and collaborators and funding sources, and thank you for listening.